Well, I am Fernando Baldi. Luis present me. I am professor at the UNESP. It's the other university of São Paulo State. And also I am involved with industry. The UNESP is an association of research and breeders aiming to provide genomic evaluation for the farmers. The topic of my talk is about the impact of genomics to improve the beef productions in the tropics. Here are the main, I list the main topics or concerns about beef production in the tropics. As we know, in the case of Cebu breeds, we need to improve growth and sexual precocity because are traditional important traits in order to improve productivity. We also concern about improved beef quality, particularly in Cebu breeds, and we are concerned also with feed efficiency. And as we show, we are working with new traits related to environment impact of beef production, and also I will show some preliminary results about multiomics integration in order to improve not only genomic evaluation, but also use this information for management decision. In the case of UNESP, the UNESP started the genomic breed evaluation in 2013, first using the multi-step genomic evaluation in a partnership with Tropics, similar to like Marcus showed. And since 2017, we are running the single-step genomic lab. And nowadays, most of the Cebu breeding beef cattle programs run genomic evaluation. Today in Brazil, there are many breeding programs here in Brazil, particularly for Nelore breeds. And the measure impact of genomic selection is the widespread use of young sires, as I will show. In the case of UNESP, the UNESP provides several GVBs for Nelore, Guzerá, Brahman, and Tabapá. That are the main breeds for beef production. And in the case of UNESP, has growth traits, female reproductive traits, like scrotal circumference, maternal ability, that are the traditional GVBs for beef cattle. And also recently we incorporate feed efficiency, like residual feed intake, and also traits related to beef quality, like beef tenderness. Here we are going to show the impact, okay, of genomic selection. As we know, the major impact in genomic selection is in young animals, but it's important to note that the trait that benefits from the genomic selection is reproductive traits and maternal traits that from the economical point of view are very important traits for beef cattle production. Here are the results from one Nelore herd, the Bella Alborada, showing here the GVB trends for weaning weight and for cow accumulated productivity. That is an important, economically important trait. And here in 2013, we implement genomic selection, and the impact of genomic selection in the genetic trend was really very important, showing for example, for growth traits, the genetic trends increased 3 times, and in the case of cow accumulated productivity, the genetic trends increased close to 4 times. That is very important because not also the genomic selection provides better or higher accuracy in the young animal, but also the very important economical traits in this cattle has the higher benefit. Here, this slide is showing the impact of using genotype young sires. The example here is for ribeye area. That is an important trait for beef production. Here we have the size ranking in function of the size per year, showing that the young sires have better genomic evaluation. And here in the right, we have the phenotypic trend, okay, showing that in the last 15 years, that is almost the same time period when the genomic selection was implemented at the NECP, showing that we have a great impact, okay, in the carcass yield and also in the carcass weight. That seems a small impact, 10 kilograms of carcass weight by animal. This is the increase, but we transfer this increment, considering that in Brazil there are close to 7 million of heads in feedlots. The potential that animal breeding and particular genomic to increase the output is really very high. Okay. Here is another trait that some beef cattle breeding programs here in Brazil deliver genomic EBD for residual feed intake. That is a trait related to environmental impact. Here we classify the top-ranked GVB site for residual feed intake, and here the bottom-ranked pool. And here in the figure we have the residual feed intake of the progeny, and we have between the two groups a difference of close to 300 grams of dry matter intake per day. Okay, but this small number, if we use this number, for example, or if we extrapolate this number for a feedlot with 10,000 animals during a period of 150 days of feedlot, we expect that we need less corn or less feed in order to supply in the feedlot. This is an interesting result showing the impact of selecting for feed efficiency, particularly for residual feed intake. Here in the left side we have the dry matter, the adjusted dry matter intake in the wild animals, in the animal that has dry matter intake records obtained in the growth safe system. We know that the dry matter intake was almost the same during the last 10 years. And here in the right we have the same trait, the dry matter intake, the predicted dry matter intake, but for a herd that incorporate the residual feed intake as selection criteria. This is the result from Rancho da Machina. That was the first farm here in Brazil that used growth safe to obtain residual feed intake. And we can note that the dry matter intake in the last 10 years decreased about 10%. This is an opportunity to, for example, to increase the stocking rate, particularly in beef cattle systems in Brazil, that most of the beef cattle systems perform in pasture. So there is an opportunity, okay, to decrease in input and also to increase the output, increasing the productivity. Okay, as I suppose that all know that the main beef cattle breed in Brazil is Nelore, but there are also small breeds like Guzerá and Brahman and also Tavapuá. And the main limitation to perform genomic selection in this small breed is the small reference population. This is an ongoing study performed by Marisol, showing the advantage of using multi-trait genomic evaluation using metafounders. Here we have the result of single breed evaluation for different traits. And here we have the result of genomic prediction using multi-trait. As we can see, the accuracy for all the traits increase when we perform genomic selection using multi-trait, multi-breed evaluation. And there was higher impact on breed with small reference population. because here we run Gusera, we run a genomic evaluation using Gusera, Brahman, and also Nelore Cat that has the largest genotype and phenotypic database. Okay, we also perform some studies because genomic selection allows to select for difficult to select traits like beef fatty acid profile in Nelore. Still, we don't have GBB for this trait, but in the case of beef tenderness, the NECP provides GBB, okay, for beef tenderness. In the case of these are the GBBs for beef tenderness in Nelore cattle. So the farmers and the industry has the possibility to select or to identify Nelore side with higher beef tenderness. We also are working with new phenotypes like optimal slaughter point. This phenotype is obtaining through automatic weighing and also using images. And through this information, the software and the artificial intelligence estimate the optimal slaughter point in the animals. This is the photo. Here are the cameras, and here's is the weighing. And also we estimate that there is genetic variation for this trait. And here in the graph we have the top ranked sire for GBB profitability in the feedlot. And we can see here in red the GBB, and in blue is the progeny record for profitability. These are the top ranked bulls. These are the bottom ranked bulls. Okay, and we compare the profitability records. Progeny, the top sires evaluated for profitability produce progenies that have a higher profitability in the feedlot. We also we think that in the future the multiomics application will allow to understand and predict better using big data analysis. And I think that integrating not only genomics, but also the other omics like metabolics, proteomics, we will help to identify biomarkers in order to include these biomarkers in the genomic evaluation, or in commercial farms to use this information to improve the management decision. In this sense, these two works are ongoing research. Here, we estimate the prediction ability for early heifer pregnancy, including quantitative trait nucleotides. Here, we include ETLs close to, we weighted, differentially weighting SNP close to KTLs associated with heifer pregnancy. Okay, the accuracy and also the accuracy increase and the inflation reduce when we include external information in order to improve the genomic evaluation. This is an ongoing research, but in this case, with residual feed intake, that with differential weighting, SNPs close to a SNP that has an effect on gene expression related to residual feed intake, and also the prediction ability, and particularly the prediction inflations improve when external information was incorporated in the genomic evaluation. Okay. Another application of genomics is to use genomics to reduce inbreeding and improve the mating decision. This is a paper produced by Elisa. Here, this is for Nelore. Here we have the traditional inbreeding, okay, that was almost constant. And here we have the same animal, but using genomic inbreeding, okay? That genomic inbreeding, you can capture better linkage between animals, and also you have a better estimate of genomic. And this information can use to improve mating software, and also to maintain the genetic variability. Okay, in order to finish, okay, this is some topics about future perspective for incorporation genomic information in beef cattle evaluations in Brazil. In Brazil, it's very common the use of F1 animals that result from the crossbreeding between Angus and Nelore. And there is an opportunity to use genomic information to evaluate F1 animals, aiming to produce higher beef quality and also to improve efficiency in feedlot conditions. Also, there is a concern of using genomic information and also to use altered traits related to environment impact. The topic about genotype environment interaction is also an interesting topic, particularly in tropical conditions, because in tropical conditions there are large variability in terms of environmental conditions. I think there are some opportunities to use genomic information to improve particularly mating decision and management decision. The topic about lowering the cost of genomic information, particularly for commercial farms, is an interesting topic, because today the main limitation to apply genomic information, particularly in commercial farms, is the cost of technology. That is an important topic to develop cheaper technologies. And I think that is also important is to develop models and softwares in order to integrate not only genomic information, but also phenomics information in order to improve the genomic evaluation and also to improve mating decision or management decision. Okay, I would like to thank all our partnerships, and thanks for the attention.